Latent fingerprints are those invisible prints left on a surface through the natural secretions from our skin. Unlike patent prints, visible prints, and plastic prints, impressions left in soft materials, latent prints require special techniques to be visualized and analyzed. Understanding the chemical composition of these residues is crucial for effective detection and development. Composition of fingerprint residue. Latent fingerprint residue is primarily composed of various substances secreted by the skin. These substances can be broadly categorized into three main types. Sebaceous secretions, produced by sebaceous glands, these secretions are oily and waxy in nature. Eccrine secretions, produced by eccrine sweat glands, these secretions are primarily water-based and contain various dissolved substances. Exogenous substances, these are external contaminants that can be picked up by the fingers, such as dirt, oils, cosmetics, and other environmental substances. Let's break down each category further. Sebaceous secretions. Sebaceous glands are found primarily on the face, scalp, and upper body, but their secretions can be transferred to the fingertips through contact with these areas. The main components of sebaceous secretions include lipids. These include fatty acids, triglycerides, wax esters, and squalene. Lipids are hydrophobic, making them crucial for certain fingerprint development techniques that rely on non-water-soluble substances. Cholesterol and its derivatives. These compounds can also be found in sebaceous secretions and play a role in the overall chemical profile of the residue. Eccrine secretions. Eccrine sweat glands are distributed across most of the body's surface, including the fingertips. The primary components of eccrine secretions include water. Constitutes about 98 to 99% of eccrine sweat. Evaporates quickly but leaves behind other dissolved substances. Inorganic salts. Sodium chloride, NaCl, common table salt, which can crystallize upon evaporation. Potassium, K+, present in smaller amounts compared to sodium. Calcium, Ca2+, and magnesium, Mg2+, trace elements that can influence the residue's chemical properties. Amino acids. Serine, one of the most abundant amino acids in sweat. Glycine, alanine, and glutamine, other common amino acids found in sweat. Cysteine and tyrosine, present in smaller amounts, can react with ninhydrin to form colored compounds. Proteins and peptides. Lysozyme, an enzyme with antibacterial properties. Dermcidin, an antimicrobial peptide. Other minor proteins and peptides, present in trace amounts, contributing to the residue's chemical profile. Urea and ammonia. Urea, a waste product of protein metabolism. Ammonia, also a metabolic byproduct, contributing to the pH of sweat. Lactate. Lactic acid, a byproduct of anaerobic metabolism, contributing to the acidity of sweat. Lipids. Fatty acids, small amounts present despite the primarily watery nature of eccrine sweat. Sterols, such as cholesterol, though in very low concentrations. Glucose. Sugar, present in small amounts, can be significant for individuals with diabetes. Exogenous substances. Exogenous substances are external materials that come into contact with the skin and can be transferred to a surface through fingerprints. Here are some additional examples. Environmental pollutants. Dust and dirt, particulate matter that can be picked up from surfaces and environments. Soot, common in industrial areas or from handling burnt materials. Pollen, can adhere to fingers from touching plants or surfaces with pollen. Food residues. Oils and greases, from cooking or handling oily foods. Sugars and starches, from handling fruits, candies, or other sugary foods. Proteins, from meats, eggs, or dairy products. Personal care products. Lotions and creams, moisturizers and other skincare products. Makeup, foundation, powder, and other cosmetic products. Soaps and detergents, residues from hand washing or cleaning products. Occupational contaminants. Grease and oil, from working with machinery or automotive parts. Chemicals, various substances depending on the profession, such as paints, solvents, or adhesives. Metallic residues, from handling tools or working with metals. Textile fibers. Natural fibers, from cotton, wool, or other natural fabrics. Synthetic fibers, from polyester, nylon, or other synthetic materials. Understanding the detailed composition of exogenous substances and Eccrine secretions is essential for forensic experts in the development and analysis of latent fingerprints. Each component plays a crucial role in determining the most effective technique for visualizing and interpreting these invisible prints.